So my lab is focused on trying to understand X chromosome inactivation. And as a mascot, we use the calico cat because it's a visual representation of X chromosome inactivation. In X inactivation, the inactive X chromosome acquires what we consider all the classic marks of heterochromatin. It becomes late replicating, acquires repressive histone modifications, becomes hypermethylated at CPG islands, goes to the nuclear or nucleolar periphery. And we want to understand how that happens. The initiating factor for X inactivation is actually expression of the long non-coding RNA exist. And I discovered the long non-coding RNA exist during my doctoral studies with Hunt Willard. And in the 25 years that I've had my lab at the University of British Columbia, we've continued to try and understand how X inactivation occurs. So for EXIST, what we'd like to know is how does a non-coding RNA spread along a chromosome, and why does that spreading recruit all of those repressive modifications? We know that the 17 kilobase RNA actually has different modules which recruit different interacting proteins. And in mouse, it's been fairly well studied what those proteins are doing, but we study humans. And the EXIST RNA has many similar regions between humans and mice, but also a number of regions that have diverged and amplified different domains. And so not surprisingly, we see that the process in humans has some important differences. In addition, another big difference between humans and mice is in the number of genes that are subject to inactivation. So in humans, approximately 25% of the genes continue to be expressed, at least partially, from the inactive X chromosome. About half of those are consistently expressed in all tissues and in all females, and the other half are what we call variable escapers. They differ between females or between cells or between tissues in whether or not they will escape inactivation. So I think the rapid increase in epigenome-wide studies has allowed us to identify many of the factors that are recruited to an inactive X chromosome. I think we're beginning, particularly for those researchers who study mice, to get an understanding of the process by which these marks are acquired. And in terms of the escapees, I'm quite excited by our recent studies, which has shown that if we put a transgene of human DNA containing an escape gene into mice, we can see that the mice are able to recognize the DNA elements and allow that gene to escape in mice, despite the fact that the mouse ortholog of that gene is subject to X inactivation. So I hope that means that we'll be able to identify the DNA elements that are enabling not only spread of inactivation, but also the ability to not be silenced, despite it being a chromosome-wide process. And of course, the big thing is that we want to understand what's the impact on human health and disease. And more studies are now including sex differences in their studies. And I think it's really intriguing that the human genes that escape inactivation actually end up being expressed more in females than in males. Many of them are, in fact, chromatin regulators or transcription factors, so they can be creating an environment where there's differential expression. And I think that can be contributing to the sex differences that we see in disease susceptibilities.